Hey guys, alright, snow day number 3, and I couldn't think of a better day than to start our 11-3 notes on the video. I'm at home, I got my Rainbow Bright Snuggie on, I'm ready to go. If you've never heard of Rainbow Bright, popular cartoon when I was growing up, you can look it up. But alright, 11-3 talks about independent assortment, and if you break that down, independent means it works on its own, and when you have an assortment, you usually have different things that you're trying to organize or to assort in one way or another. So what we're relating this to is what happens if there's more than one gene. So we know we have an assortment of genes in our human genome, but does inheriting a certain gene for seed color affect the inheritance of another trait like plant height? So we want to know if these genes actually independently assort, or is one affected by the other. So we can do this by performing two-factor crosses. They're also known as dihybrid crosses. Remember that di means two, hybrid means a mix. So Mendel performed experiments to follow two different genes as they pass from one generation to the next. And we're going to do an example here in a second. He crossed plants that were true breeding for two different traits. So he did round yellow peas, and those had the genotype of big R, because round is dominant, and big Y, yellow is dominant. And then wrinkled green peas. So the little r designates wrinkled, the little y designates green for the genotype. And that just answered the question below, which traits are dominant, which traits are recessive. Round and yellow are dominant, they're represented by the capital letters. Wrinkled and green are recessive, they're represented by the lowercase letters. Alright, so this is a dihybrid cross, and I know it looks really complicated, and I'm going to try to use my drawing tools to help you guys out with this. It's probably not going to be the best, but we'll work with it. So the way that you figure out the gametes that are possible over here from mom and dad, so we'll just, this is mom, this is dad, is you take one at a time, kind of like foiling in math. So if I take big R, or I'm sorry, it's a little r, and this little y, I add it right here. And then I could also have this little r and this y, and that we'll just say it's this one right here. And I also need to take this y to both r's, and so I'll just say that one is here, and this y to the other r, which you get this combo right here. And I know, voila, it's the same every time. But as you'll see in the following example, it's going to get a little bit more complicated. But basically, foil, you get every combination possible. You would have done the same thing with the homozygous dominant genes. Okay, and again, you're going to get all the same. So guess what? All the offspring are also going to be the same. So when you're doing the dihybrid cross, you follow the same way. You go over and up. And so we have big R and little r, and then we have a big Y and little y. Notice that I kept the r's together and the y's together. You should keep the same alleles for the same gene together. Okay? So, and again, that will be the same for the whole thing, since all of the gametes that are possible are the same. And we're going to carry this problem on onto the next slide. So, why are there so many boxes in that dihybrid cross? Well, each parent can produce 4 different sex cells. Okay, remember sex cells are called gametes. And each gamete has an equal chance of combining with each of the other parents 4 types of gametes. So therefore, we have 4 options as it's showing you in the picture over here. So our F2, which is our second set of offspring, dihybrid cross. Each of the offspring in that example we just did became hybrids for both traits. Again, dihybrids. So Mendel crossed these offspring to produce another generation of plants, or again, the second offspring is designated by that F2. So if the genotype of each parent is big R, big Y, little y, what kinds of gametes will each parent produce? Okay, so here we are going to get the gametes from each parent, and remember you're going to use that foiling technique that I just showed you. So if we take big R and big Y, that could be our first set of gametes. so you would write your first one here. And then if you take big R and combine it with little y, that would be another option. Then you need to take little r and combine it with big Y. Again, don't mind my writing. It's really hard to use this tool and write. Your last option would have little r and little y. So, 4 sets of gametes possible from the parents. And since parent 2 is the same as parent 1, I'm not going to go through all that. It's done the exact same way. Okay, so now basically we figured out the gametes for these parents, so we can just write them up here. So we had big R, big Y, big R, little y, little r, big Y, and little r, little y. Same for our parent over here. Again, they are both the same. They were going to give the options for the gametes are the same. Big R, big Y, big R, little y, little r, big Y, little r, little y. And now you just perform your cross, again, like I showed you earlier. So in this case, if we go over and up, we have a big R and a big R. So big R, big R, big Y, big Y. Go over and up to this square, we still have big R, big R. This time it is big Y, little y. Over and up, we have big R, little r. And big Y, big Y. And then big R, little r. Big Y, little y. Alright, and you would do this for the whole dihybrid cross. And I'm going to show you on the next page. These are the results that you would have at the very end. Okay, so you can see the ones. I think they did put them in the same order that I did, so I had the same ones up here. Notice this one visually shows you round and yellow or round and green. Wrinkled and yellow, and then your wrinkled and green. And this would be your F2 generation offspring. If you were to count these all up, what you'll notice in a dihybrid cross with parents that are dihybrid for both genes, you usually get a 9:3 to 3:1 ratio. So I have 1, 2, 3, 4, 5, 6, 7, 8, 9 that are round and yellow. 1, 2, 3 that are round and green. 1, 2, 3 that are wrinkled and yellow, and only 1, which is both recessive types, that is wrinkled and green. Okay, so the results of this cross do show us that the alleles for the 2 different traits segregate, or they separate from each other independently into the gametes. So one is not dependent on the other. And then again, like I said, the offspring in this cross will usually show or they will show, I should say, a 9:3 to 3:1 ratio of the different phenotypes. So, summing up Mendel's principles, inherited traits are determined by genes, and these genes are passed on from our parents to the offspring through our gametes. Some forms of a gene may be dominant and others may be recessive. And then the genes segregate during meiosis, so only one copy of a gene goes into the gamete, and alleles for different genes usually segregate independently of one another. And we're going to go ahead and stop there. I'll see you next time. Maybe we'll actually be at school tomorrow. Have a good day.